We'll get started. All right. Thank you. These are our disclosures for the program. My name is Dr. Janjigian. I'm a medical oncologist and chief of GI oncology at Memorial Sloan Kettering Cancer Center in New York. And I'll be your, maitre d' for tonight and the chair for this program. I'm really excited to be able to review precision tactics in HER2-positive gastrointestinal malignancies, where we are with these pan-tumor approvals with HER2-targeted agents, and of course, what the future will be for our patients. Really, I'm so lucky to be able to stand up here with experts in the field of GI malignancies, Dr. Shubham Pant, who's a medical oncologist and leads the early phase development and treatment options at MD Anderson, and Dr. Strickler, who's also an expert in targeted therapies in colorectal cancer at Duke, and also is really focused on circulating tumor DNA-based therapeutic development. So really thank you for my colleagues for joining us on such a busy night at ASCO. I know there's a lot of interest in this field, and we have many participants online. So when you're speaking in the Q&A session, please wait for the mic and so that our online participants can queue in as well. So thank you, of course, peer review and the Cholangiocarcinoma Foundation and the Colorectal Cancer Alliance for making this possible, as well as our sponsors from AstraZeneca, Daiichi Sankyo, for providing this educational CME grant for the symposium. If you haven't already done so, please complete the pre-event survey, and reminder to look out for additional follow-up polling questions during the treatment. We know that the best way to learn about the new information is to test yourself and quiz yourself, and that's what we're doing for you. And please submit your questions online. I have a iPad here, and we will monitor the questions. Probably as you think of them, just jot them down, and then I'll direct them towards speakers in the Q&A. And then, of course, all of the information you will be seeing today is available for download on peerreview.com 2024 GI. So you can really follow it along, download it, save it for your fellows, trainees, and so forth, NPs, and et cetera. And of course, join us on Twitter and feel free to tweet and voice your opinions. You can tell us we absolutely disagree with everything the experts say, and we can start a debate. So our goals for today is really to improve your understanding of the biologic rationale for HER2-targeted therapies, both for protein overexpression, amplification, and mutations, and review the latest evidence supporting these biomarkers for pan-tumor approval for HER2-targeted therapies, particularly as it comes to GI. We will help you gain the skills to implement personalized team-driven treatment for your patients with HER2-targeted approaches based on safety, efficacy, really get the real-world feel for how to use these agents in the clinic, what to watch out for in practice with side effects related to these treatments, and what are the guidelines recommending for patient-specific needs. And also provide you with the tools to address practical aspects of the care of HER2-targeted therapies, how to lean on your multidisciplinary team to help you manage and to get these patients through more therapies and coordinate the care. So of course, our partners in this are patients and patient advocates and their caregivers. So the Cholangiocarcinoma Foundation is an excellent resource for professionals, patients, and caregivers. It's an advocacy group with a mission to find, cure, and improve quality of life for those affected with cholangiocarcinoma. Bile duct cancers certainly have been front and center for targeted therapies, and you can find new resources at cholangiocarcinoma.org for multiple these patient and caregiver-facing resources to help connect patients with each other and to educate them, and certainly to help them find clinical trials and to participate and enroll in those trials. And of course, there's many other resources and tools available, and if you peruse peerreview.com, you can download these practice aids. Colorectal Cancer Alliance resource is also critical to our colon cancer patients. It empowers a nation of passionate and determined allies, and I've met several of these individuals over the few years, and certainly it's been amazing the advocacy and the voice this foundation gives to our patients with colorectal cancer. So please visit the QR codes below, and downloadable practice aids are also available to learn more at colorectalcancer.org. Help the patients navigate the clinical trial portfolios, and you can also learn more about screening. And as always, you can download these resources as well and see what other practice aids are available on peerreview.com. So with that introduction and overview, I will begin with just a broad sort of landscape of HER2 and how we think about it to help personalize therapy. For, across solid tumors, you could see it is a pan-tumor agnostic biomarker. Certainly in breast cancer, it's been known for many, many years and has been a validated biomarker. And what we're learning now is that approaches that may have worked in one disease may work in other solid tumors, particularly in GI tumors, with some modification because of difference in co-occurring alterations, different pattern of expression. And here we have a way of thinking about it. In purple, you have, the mutation-based alterations, which is what we typically think about in lung cancer, right? HER2 in EGFR mutations are known drivers in that disease. And then amplification and overexpression is how we typically, for example, think about it more in breast and stomach cancer as well. But even within those, really recognizing that, yes, it's an agnostic pan-tumor biomarker, but how do we improve and personalize therapies for this biomarker based on patient-specific disease characteristics and biology for an organ site? And I can teach you about my experiences with this in gastric cancer, and our panelists will also cover colon and biliary tumors. So the number of HER2-targeted therapies being evaluated in GI cancers between ADCs, bispecifics, CAR-Ts is really just the fun just starts. So I think it's very important to learn about this and to keep an eye on developing clinical trials so you can offer the patients the best options. So the real-world evidence really suggests that HER2 is an unmet need. We are seeing more and more patients getting tested for HER2 in first-line setting, particularly in GI tumors. But we can definitely do more. I mean, real-world data suggests that even in disease where HER2 has been validated as a biomarker for over a decade, which is gastric cancer, not every patient is getting HER2 testing, only about probably 50 to 60%. And so we definitely can improve on that. It is typically a negative prognostic biomarker outside of HER2-directed therapy. In breast, at least. In gastric, the data, the patient's survival is relatively equivocal because it's a more aggressive disease biology. As you know, HER2 is a transmembrane receptor that is a member of 4 receptor superfamily, and EGFR is an important one, and certainly the dimerization partners such as HER3 and also HER4. And these receptors share common variants and structural variants. And what the issue is in GI tumors particularly is that we also have activation of downstream effects such as MAP kinase, RAS, PI3 kinase, and so forth, and parallel pathways such as EGFR. And so that's what I was alluding to, that it's not as simple as in breast cancer, or not to say that breast cancer is simple, but it's not just a cut and paste approach. In GI tumors, we really need to understand. And these are some of the targeted agents that are already in the clinic, right? So we have ADCs, antibody-drug conjugates, we'll talk about a bit, and there's different drug-to-antibody ratios, different payloads that we talk about. There's biparatopic antibodies, such as zanidatamab, that bind different epitopes on HER2, and also pertuzumab. And there's certainly bispecific antibodies that are designed to really grab both HER2 with other partnering agents, because we know, for example, in gastric cancer, HER2 often occurs with other immune checkpoint targets such as PD-L1. The tyrosine kinase inhibitor story is still evolving, especially in colon cancer. It seems to be very effective. And then whether or not you need irreversible or reversible inhibitors are okay, such as tucatinib, is still to be determined. So it's just a little appetizer on HER2, and now we'll switch gears and talk about, I guess, the specific characterization of gastric cancer, because after breast, that's where most of the data right now is, and really where are we with the modern strategies with these targeted therapies. So the typical workup in the clinic involves, and so the TOGA study obviously came out in 2009, and since 2009, HER2 has been a validated biomarker. We typically prefer to test it in first-line setting, and depending on where in the GI cancer, the rate of HER2 positivity varies slightly. So for example, in the proximal stomach, it's 30%, while in a distal stomach, it's 15 to 20%. And so that affects the variation in incidence across the different clinical trials, East versus West, and so forth. We know that immunohistochemistry by IHC is the preferred biomarker first, and then for IHC 2+ equivocal cases, we like to do fluorescent in situ hybridization, looking at the ratio of the chromosome to the centromere, and anything above 2.0 is considered HER2 positive. So in first line, that's what we do. And for a while after approval of trastuzumab, there was a lull and a lot of negative studies, and we'll talk a little bit about why that is. And in second line, there were one after another negative study until trastuzumab deruxtecan really changed practice, and we'll talk a little bit about that as well. But the reality in gastric, unlike breast cancer, globally, if you look at phase 3 data, only about 50%, if I'm being very generous, 50% of patients get on to second-line therapy, including HER2 patients. So that's really an important factor to consider as we're taking care of patients, monitoring their cancer-related symptoms, addressing them head on, and also maybe imaging them and following them for toxicity to really change therapy before deterioration of clinical symptoms so that you are able to treat more patients with second and third line therapy. So this is kind of the typical epithelial staining that you see on the left with a tumor heterogeneity and also incomplete membranous staining. Gastric epithelium, obviously, because of its nature and chronic inflammation and the secretory nature of epithelium, is different than the breast epithelium. So there was in 2009, when the TOGA study first came out, there was a big learning curve for the pathologists, especially in non-specialized cancer centers who are focused in breast, to then change gears and understand how they should score the testing in gastric. I think we figured it out. Basically, it's a relatively low threshold, right? Anything, even if you have 5% or 10% of focal positivity that's strongly positive by HER2 IHC, the pathologist will call it positive. And that's where the issue comes in because of heterogeneity, because within each tumor sample, and certainly with different metastasis in each patient, you may have these variation in levels of HER2 but with focal staining with IHC, you just won't understand how likely is this patient going to really benefit from therapy. So testing, is really important. I think I don't want to make it too complicated. You should just test everyone. HER2 is an important biomarker. We test it definitely in first-line setting. We're testing it in locally advanced resected tumors as well, because more than 50% of those patients will recur. And again, we start at IHC 3+, and then if they're 2+, then we do FISH. And that's supported with all the guidelines across the world. Why do we start with IHC as opposed to FISH? Well, we know from the TOGA data that patients that are IHC 0 FISH positive don't benefit as much. It's a different, and it's likely because the FISH positivity is really, either really not, it's, not a true amplification of the gene as opposed to just duplication of the actual chromosome, or if it's so focal that it was just not enough to make the protein overexpression. So, if you look at the modern data, for example, Keynote 811, the comparator arm does better than the TOGA, arm. And people ask me that all the time. Why is the reason? Well, it's because we've changed the way we select patients. We select them by IHC 3+, IHC 2+, as opposed to in TOGA where FISH entry was the criteria. Of course, in breast cancer, now HER2 intermediate or HER2 low is the, everything, is sort of glass half empty, glass half full approach. So in gastric, we're moving toward that as well. And there is data to suggest that you may be able to target HER2 intermediate cases as well. So there's several studies looking at combination therapies with T-DXd-based regimens in HER2 low. But right now, the level 1 sort of FDA-approved biomarker that's in all package inserts is IHC 3+ or IHC 2+ FISH positive. We don't typically FISH IHC 0 or 1+, but again, it's on case-by-case basis. If you have a good pathologist who thinks they should be, if there's maybe borderline overexpression, it's reasonable. But those patients just don't benefit from trastuzumab-directed therapy. So what are the options? After a decade of negative studies, finally we broke sort of the ceiling in the first-line setting. And now, in addition to trastuzumab monotherapy with chemotherapy, the preferred regimen is combination of trastuzumab and pembrolizumab, because particularly almost 90% of those tumors are also PD-L1 positive, and we really demonstrated a significant improvement in all outcomes in survival, including response rate and progression-free survival in first-line setting. Trastuzumab deruxtecan resurrected the whole concept of targeting HER2 beyond progression in trastuzumab progression in second-line therapy. So T-DXd is also approved at 6.4 mg per kg in US and Europe and Japan for use in second-line. Actually, in Japan, it's in third-line still, but in US and Europe, it's in second-line, with a caveat that if safe, we would definitely recommend, and it actually says that in the FDA insert, package insert, to biopsy and confirm progression, HER2 positivity at progression. So let's review again the data. So the TOGA regimen, as I mentioned, this was the first study. There was the first biomarker study in gastric cancer, and this was published in 2010, and it demonstrated survival benefit for addition of trastuzumab chemotherapy with chemotherapy, and this was usually 5-FU cisplatin-based combination compared to chemotherapy. As you can see here, the curves separate relatively early with a sustained separation on the curve, and these curves are for all comers. Hazard ratio is 0.74. If you look at the data for the patients that you and I would consider HER2 positive in the clinic, which is IHC 3+, IHC 2+, FISH positive, the data looks even better. So the subgroup analysis does suggest, as you can see here, that the patients that benefit the most are the HER2 IHC 3+ patients that are also FISH positive, and that really, IHC 2+ FISH positive as well, although it's a rare, smaller subgroup. In the modern studies, it's probably only about 30% of the patients, so the confidence intervals are wide. And the patients that are FISH positive, IHC 0, not so much. They don't really benefit, and again, for all the caveats that we talked about. Data that we didn't include here, but it's also been published, and you can look it up in its 2018 Cancer Discovery paper, is that on sequencing, because that's a question that comes up a lot, on NGS, for example, if you have a clinically HER2 positive case by IHC or a FISH, and then you sequence it, and you don't have CTD, HER2 amplification by sequencing, usually what it means is probably insufficient DNA content, or it was such a focal positivity that it didn't meet the threshold for HER2 positivity. And we have data from our center showing that those patients, their progression-free survival on first-line therapy is quite short on first-line trastuzumab. So the challenge with HER2 in gastric is that, as I mentioned, it co-occurs with other things. Unlike breast, GE junction tumors in particular, which is the highest percent of HER2 positive tumors, 80% are P53 mutant, they're chromosomally unstable, they have high level of aneuploidy, and they have other co-occurring alterations. So targeting HER2 with just better HER2 inhibitors, such as trastuzumab lapatinib or pertuzumab-based combinations, just did not work. And we showed that in gastric cancer in particular, very high level of PD-L1 overexpression co-occurs with HER2. And in fact, as the patient is on trastuzumab, the PD-L1 level goes up. So that led to our preclinical data, and then clinical phase 2 data, and then phase 3 data. So it took about 5 years to get this combination to the clinic, but now it's in the clinic, so please use it. It's combination of pembrolizumab, trastuzumab with chemotherapy compared to the TOGA regimen, which is with the only caveat where IHC3 plus, IHC2 plus FISH positive patients were treated. And instead of cisplatin, we used oxaliplatin. And with that, the control arm did actually better than you would expect. The overall response rate with TOGA is about 47%. Here we did 52. Overall response rate is 74%, higher percent of complete response rate, and the duration of response and so forth. So just by first interim analysis, looking at response, because we were expecting very high response, we did a phase 2 at MSK where the response rate in a single arm phase 2 was 91% with 100% disease control rate. So we expected high response rate with some attrition accounting from phase 2 to phase 3 transition. And so we put it interim analysis as the interim look, and it got it approved just based on that delta. And since then, we were able to present and publish progression-free survival. As you can see here, the intent to treat population on the left and CPS greater than 1 population on the right. There's clear benefit with median PFS of 10 months in the all-comer population. The survival curves separate early and with sustained separation. And you probably saw the press release for the final analysis, and we did meet. So this data will be at ESMO, so you can stay tuned for that. It is nice to see these waterfall plots. It shows that this regimen works. We also saw a higher percentage of patients with more than 80% reduction in their disease volume. So clinically, this is meaningful because our patients present quite sick with symptomatic disease, inability to eat, and so forth. So, targeting immune checkpoint access together with HER2 really helped, while targeting HER2 just in a more thorough way with TKIs and antibodies did not work, which is slightly unlike the story in MSS colon, which John is going to tell you about. So this is actually good. We put in the curve after all. This is the curves I was telling you about from the cancer discovery paper. It was a very small, sort of hypothesis-generating analysis. We just sequenced in our first-line setting 50 patients with HER2-positive tumors undergoing standard of care chemotherapy, trastuzumab. And as you can see here, patients who have no ERBB2 amplification on IMPACT, which is our 500 tumor-matched panel, they don't benefit. They behave as if they were HER2 negative. And then if you're on that gold line, where you have high level, top quartile level of HER2 by NGS, and you have relatively RTK quiet tumor, meaning you don't have KRAS amplification or PI3 kinase mutation, or MET amplification, MYC amplification, all these other things that these tumors have, then you tend to do even better. So it really helps understanding these patients and how they're going to do. We have similar biomarker data from our phase 2 pembro-tras paper. And what's interesting, with addition of pembrolizumab, HER2 is still the important driver of response because of the ADCC and ADCP related to trastuzumab. So what about after the patient progresses? And when I first started as a GI oncologist, actually, my first set of trials were all focused in second-line setting. And it was before TDXD, of course. But what was really discouraging is that what we see in first line, which is what I was telling you about, heterogeneity, aneuploidy, activation of downstream MAP kinase signaling, just gets worse the longer the patient stays on HER2-directed therapy. So targeting it in second line with TKIs and other things that we usually do it with is even harder, and the duration of response is very, very short, like measured in 3 to 4 months. This was the patient that we biopsied to go on one of our irreversible TKI studies, and he had a loss of HER2 with really robust initial response to trastuzumab and now progression. And so you can see here at the top panel, there's a really nice brown staining with uniform HER2 IHC overexpression, and it went from pretty much 3 plus to 0. So that was very sobering. And again, when we were designing the phase 2 TDXD studies, we really made it clear that they had to biopsy, and thankfully we did, because it really is important to confirm the biomarker actually exists before you target it. And this oncoprint does show that the level of EGFR, level of RAS, PI3 kinase activation just gets worse over time. So with HER2, I think targeting it, again, with off-target effects, which we'll talk about with the ADC assets, but also in the first-line setting before the tumor gets this, aggressive, and debulking it with other agents is really useful. So what is different about TDXD compared to, for example, TDM1? Because TDM1 came first and did not improve outcome in second-line gastric compared to paclitaxel. This was unfortunate, and it didn't help our patients, and kind of also set back the field. Well, so TDXD studies, first of all, biopsied patients before they went on, at least in the United States. And second of all, when we talk about ADCs, we always care about two things. Obviously, what's the target, HER2? What's the drug-to-antibody ratio? And it's quite high here, it's 1 to 8. And then what's the payload, which is here we have, unlike a microtubule payload in TDM1, we have a topoisomerase 1 inhibitor, DXd, which is very, very active, obviously, in GI tumors. Irinotecan is very helpful in our diseases, and we know how to give it, and have a lot of experience. So the data for TDXd first came in third-line setting, and so we aggregated here Destiny Gastric 01 and 02 studies, looking both at second and third-line setting. The one at the bottom is the one that was published in New England Journal of Medicine that looked at, it was a randomized study looking at TDXD versus investigator choice chemotherapy, which is usually, again, as I mentioned, involves taxane of some sort or irinotecan. And this patient's population is in Japan, South Korea, and progression-free survival and the overall survival are summarized here. I mean, the median OS in this TDXD third-line study is 12.5 months. If you remember, I was quoting you PFS in the Global Keynote 811 study, which was 10 months. So you can imagine that third-line patients in Japan do quite differently than our patients. More patients get to that stage. But nonetheless, the response rate here was very similar. Overall response rate is 40% compared to overall response rate of 42% in second-line gastric 02, which was in US and Europe. You could say, well, it's in second line, shouldn't you expect a higher response rate? And maybe, but not really. I think there's a very similar patient population in whom HER2 matters still in second or third line setting. And again, with all these agents, at least in US, we know that there's attrition between second and third line therapy, so the earlier you can use it, the better. So we prefer to use it after trastuzumab progression if HER2 is still intact and overexpressed, and that's what the FDA recommends it also. And who knows, maybe someday we'll be able to get it to first line, because that's phase 3 pivotal data is now being generated. So what about adverse event profile? We'll talk about the rare but serious adverse events related to interstitial lung disease. But I think what we can't forget, and I think what patients sometimes forget, because we're so used to now using immunotherapies and other targeted agents, is that this is chemotherapy. So you are going to get chemotherapy side effects. And the most common side effect my patients experience is nausea. People are so worried about ILD, but really it's not, it's very, very rare. But dealing with nausea in a nutritionally compromised patient who may not be all that well is really the main issue. And then, of course, as usual, bone marrow suppression. And it's interesting, depending on what line of therapy you use it in, second line or third line, I think in our patients, maybe GI symptoms are more common, and in third line patients, for example, in Asia, bone marrow suppression, because this is more heavily pretreated. They got more chemo, their bone marrow has less reserve. So what is the current status? It's used typically in second or third line setting. We actually wrote Destiny Gastric 04 to be in the United States as well. And of course, when FDA approved a TDXD in second line setting, we're now, this trial is being accrued ex-US. So our patients are able to get TDXD off trial, as standard of care, and then the rest of the world, even Japan, still has to wait for the Destiny Gastric 04 to read out. Also at ESMO, Destiny Gastric 03 will be presented, which is our study to try to combine TDXD with capecitabine or fluoropyrimidine and immunotherapy. And spoiler alert, it's now going into phase 3. So, come check out the data in Gastric 03. So detecting and managing TDXD, I think we need to understand and we need to impart it to our patients that it's a marathon, not a sprint, and they're coming off first-line therapy and maybe having side effects, usually neuropathy, fatigue, weight loss, and really detecting and managing TDXD-related adverse events, especially in interstitial lung disease, is important. I think before we get to that, I want to talk about nausea. And a lot of these usual approaches to TDXD, what I at least see in the, when I talk to my colleagues outside of our center, is that there's a bit of a reactionary approach. They wait for patients to develop nausea. But I think, being more proactive about nausea management with giving them multiple options to try at home. And even sometimes if this patient gets very nauseous and you had them on, let's say, delayed emesis regimen and day 2 and 3 steroids on first-line setting, just do the same with TDXD. They will have a lot of nausea because it's a known side effect. In terms of interstitial lung disease, we have this nifty, mnemonic of 5 S rules. I think in terms of the screening, it's only clinical assessments. So any dry cough, any new symptoms, pulmonary symptoms. For scanning, I don't scan anything more than just a regular interval scans for management of disease, symptoms. So usually we do anti-tumor assessments every 2 to 3 months. And of course, if the patient is having new symptoms, stop the treatment and scan them early. And really, having your radiologist be aware that the patient is on TDXD. And even though it's in the chart, sometimes they may not realize it. So it really is up to you to read the non-summary, the 2-page report to make sure there's no ground glass opacifications, or look at the scan yourself. Because sometimes these subtle grade 1 changes, grade 1 ILD, may be missed or underrepresented. And that's when patients run into trouble. Because if you stop the treatment with grade 1, these usually, resolve. And knock on wood, you would avoid any grade 5 events. When in doubt, steroids always work and help, especially if it's a relatively mild case. And I often find it confusing, especially in our patients who may have underlying lung disease or had COVID, differentiating between the different patterns of lung inflammation. And so have a friendly pulmonologist and the radiologist, and we often get on a 3-way call or email chain to try to figure out for each patient whether or not to stop or hold treatment. Just remember, it's palliative therapy, right? It's palliative treatment. So when it's in doubt, just hold the dose and repeat imaging. So the grade 1, I would hold until it resolved to grade 0, and then, we generally don't need steroids on them because usually they go away on their own. And if I was to resume it, I always have them see pulmonology, and we determine the risk-benefit ratio because, again, TDXD works really well in our disease, and there's not a lot of options after second-line therapy. If it's grade 2 to 4, certainly stop the treatment. I think there's something to be said about dosing because 6.4 mg/kg is the dose that's approved in gastric. I think starting with 6.4, but then preemptively dose reducing if they're having other adverse events at least in my practice, usually helps you stay out of ILD trouble. So in summary, these are our NCCN guidelines. Basically, your take-home message is please test for HER2 everyone and often. I've never had a patient say, I don't want to have a biopsy, because it's like going into a hike or a battle with outdated map. And then in second line, certainly TDXT is helpful. In first line, we are using pembrolizumab, trastuzumab chemotherapy, and maybe in the QA session, you guys can give me your opinions and tell me what you use in your practice. Other new agents to watch out for, zanidatamab is under development ex-US. Again, KEYNOTE-811 kind of scared everyone off from the first-line setting in the US, but it's still under development. It's biparatopic, targeting HER2, and certainly HER2 bispecifics, cellular therapies, and so forth. I think we're not going to have time for a case, but maybe we can answer any questions, and you have polling questions, right? So the practical points is for patients with HER2-positive gastroesophageal cancer to test for HER2 and follow immune-related adverse events. For HER2-positive gastroesophageal cancer, TDXT is approved in second-line setting. And the summary is really to test for HER2. First line, it's usually pembrolizumab, trastuzumab, and chemotherapy. Second line, we use biopsies to guide HER2-directed therapy. If they're HER2 positive, we use TDXT. If they're HER2 negative, usually paclitaxel, ramucirumab, and then I reserve TDXT to third line setting. And with that, thank you for your attention, and we'll introduce our next speaker, Dr. John Strickler. Please, John.
Yeah, thank you so much. And you've done wonderful work at Sloan Kettering, and KEYNOTE-811 is a true gift to us in the clinic who also treat upper GI cancers like myself. But I'm going to talk to you about managing HER2-positive colon cancer today. And I think the first point I would make at the very beginning is that we know that we're now in this precision cancer medicine revolution, that patients are living longer and living better with targeted therapies beyond HER2 and other targeted therapy spaces, but we are not achieving the promise of precision cancer medicine right now. And one of the reasons for that is accessing these targeted therapies. So, this dot point here on the left notes that 72% of patients are not receiving biomarker testing that completely adheres to published guidelines. I think it really kind of depends on how you define completely adhering, but the key point is here. Not everybody is getting testing that could benefit. And I think one of the challenges also is that there's no real standard protocol. It is a wild west of testing out there, and so actual biomarkers are slipping between the cracks, and that can make it difficult. And I think in general, we not only want to ensure that the testing is happening, we also need it to happen quickly, because you want to ensure that that patient is getting access to highly effective targeted therapy as quickly as possible. And we know this is important because there is this drop-off between first line to second line to third line, where unfortunately many patients are not able to access these later lines of therapy. And if we're getting the targeted, NGS or the testing too late, that patient may not even be able to access that therapy. And then there's always this practice that we still see today of recycling chemotherapy, and instead of giving targeted therapies. And so it's an educational challenge and something that I think we're working very closely with patient advocacy organizations so that patients can self-advocate and ensure that that testing is happening, it's happening quickly, and that those patients are getting access to these very important therapies. Current guidelines for testing of metastatic colorectal cancer. The key things, these are actually not all of the targets that are in our guidelines, but these are the ones that you absolutely must have as a patient diagnosed with metastatic colon cancer. KRAS and NRAS, these are well-established targets for over a decade. Certainly MSI high or deficient mismatch repair for immunotherapy. BRAF V600E is a validated target. And then one of our newest actionable targets, which I'll go into more detail on, is HER2 amplification, which is done on the same next generation sequencing panel as all of these other targets. So The good news is it's just one test to get all of these things done. So I want to focus now on HER2 in metastatic colorectal cancer. Just a few key points to take away. So where we think of HER2 as a rare target, but it's actually not that rare if you know where to look. It's typically on the left side of the colon, so starting at the splenic flexure, down the descending colon, sigmoid colon, and rectum. That's most 2/3 to 3/4 of all HER2-positive colon cancers would be over on that side. Now, Yelena just did a very elegant discussion of what HER2 staining looks like in gastric cancer and gastroesophageal cancers. It can be very heterogeneous. You can have differences in expression between simultaneous biopsies. That's not the case in colon cancer. In some ways, we think of this more like a breast cancer in terms of a HER2-positive cancer. You will see that on every biopsy specimen. You see a lot less heterogeneity than you would in an upper GI cancer. Generally, we think of this as most common in patients with KRAS and NRAS wild-type colon cancer, but about 1/4 of patients will also have a KRAS mutation, and that's relevant. I'll come back to that in a moment. Now, we think about HER2 across solid tumors as being associated with brain metastases, and that is true here of colon cancer. It's also true of upper GI cancers as well. But we also see an interesting tendency of these cancers to metastasize to lung preferentially. So, to give you a little bit more background in HER2 amplification in colon cancer, so in an all-comer population, HER2 amplification is not that common. It's maybe 2 to 3% of patients. As I mentioned, if you know where to look, that rate is much higher. So in the Heracles study in Italy, they used a diagnostic assay that I'll go into in a moment, but they found a much higher rate in patients with KRAS wild-type disease, 5.3%. Then there's been some other institutional series looking at KRAS, NRAS, and BRAF wild-type, and we see rates in the 10 to 15% range, and it may be higher in even more biomarker-selected groups of patients. So I think the key thing to know is it can be seen on the right or the left colon, but a left-sided colon cancer that's KRAS wild-type, you really do need to get that HER2 testing done. Additionally, the gold standard test is historically immunohistochemistry, which looks at protein expression, and then FISH, which looks at genomic amplification. And one of the reasons why it's our traditional gold standard is there's very high concordance between the protein staining, immunohistochemistry 3+, and our in situ hybridization for HER2 amplification. They're very concordant. You can basically bet that a IHC 3+, a high expressing HER2-positive cancer, is also going to be amplified. So those are some key points. Now, I had mentioned that there are some differences between upper GI cancers and breast cancer and colon cancer. One similarity between colon cancer and gastric cancer is that the luminal edge will not express HER2. It's only on the basolateral edge. In breast cancer, it will be completely surrounding the cell. And so we know that gastric and colon cancers just look different under a microscope, and their expression pattern is different. So one of the first challenges was just to figure out how we were even going to test for HER2 overexpression in colon cancer. So the Italians did a very nice job of building a test specifically to look for HER2 positivity in colon cancer. It was validated in nearly 1,000 patients. And make a long story short, it was beautiful work, and we never adopted it. So we usually just use gastric cancer criteria. But there are some unique challenges in HER2 as a target for colon cancer, not just how you test for it, but also there are some questions around how you would sequence it relative to first and second line chemotherapy. It's also been associated with resistance to certain targeted therapies like anti-EGFR therapies. So these have kind of created some challenges for us in colon cancer. Now, the question came up of whether this is a bad thing for a patient to have HER2-positive disease. Certainly in breast cancer before the era of anti-HER2 therapies, it was. I think the verdict is mixed for colon cancer. I would say that there's some evidence that it's associated with worse prognosis and some that it's not. And I would come out as somebody who sees a lot of this as it's probably on net a neutral biomarker for prognosis. That's my own, but it is a controversial topic. So as I mentioned, the Italians did a beautiful job of building this Heracles assay to support their Heracles study. And they used it to test the very first HER2 combination, which was lapatinib and trastuzumab. And the study was really a breakthrough for us in the field because they generated, in a group of patients where standard of care is maybe a 1% response rate, they generated a response rate upwards of 28 to 30%, and it landed in our national guidelines about 8 years ago. And so it really showed us that there was a path forward for HER2-positive colon cancer, so we owe them a debt of gratitude for bringing this target to us. Now, as I mentioned, in testing, there's this beautiful HER2 assay for colon cancer that nobody uses, and it's not even validated. It's not even used in Italy where they developed it. So how are we going to diagnose this? Because we can't treat with HER2 therapies unless we find it. I would say in the United States, our standard right now is just to put it as part of our standard next-generation sequencing panel along with all of our targets. The question is, do we miss patients? Are we going to be missing out on some HER2-positive patients if we use your MSK Impact or we use one of our commercial vendors? And the short answer is no, you will not. When you compare the gold standard test, which is IHC and FISH against tissue NGS, there's about a 93% concordance. You will very rarely miss out on a patient. And those cases where there's not concordance I suspect those are borderline positives where they wouldn't have benefited much from an anti-HER2 therapy anyway. Where you do start to see a little bit of discordances between the blood tests and the tissue tests, both IHC and FISH and tissue NGS. I'll come back to this point in a little bit. You see lower concordance at 80%. The key thing to know is you can use a blood NGS test to rule in HER2 amplification. If you see it in blood, you can trust it, you can treat it. If you don't find it in blood, you need to go back to tissue and confirm that that's HER2 non-amplified or HER2 negative in tissue. And I'll show you some data of why that's the case. So, this has been a personal journey for me. I was studying mechanisms of resistance to anti-EGFR therapies in colon cancer about 10 years ago, and working with a liquid biopsy company, one of the first ones in the field, and I was finding repeat patterns of HER2 amplification in patients. And there was that hint from Italy that the lapatinib-trastuzumab combination was active, and at the time there was a new oral, potent, highly selective tyrosine kinase inhibitor called tucatinib, which was coming in and being studied in breast cancer. So I wrote a letter to a little tiny company in Seattle, and I said, hey, we need to test this for colon cancer. We have no FDA-approved options for these patients. And they took my, first time ever somebody listened to me, and so they took my advice, and we launched this international study, Mountaineer, and this was for patients second line or later, metastatic colon cancer. We accepted HER2-positive results by any local test. And all the patients had progressed on prior 5-FU, oxaliplatin, irinotecan, and an anti-VEGF. So we initially enrolled patients in cohort A, which was the only cohort. And really, it was some beautiful data that we showed initially. So that company was bought by another company, Seagen, and they expanded it for an FDA approval. That's the randomization step, cohort B and C, with the primary endpoint being objective response rate. And this is the data we initially showed from the primary analysis. Now, I will be showing the final analysis on Monday afternoon, if you're still here. And the data, I think, remains interesting, so hopefully you'll see that. But confirmed objective response rate for cohorts A and B, the tucatinib-trastuzumab combination at 38%, duration of response over a year at 12.4 months, progression-free survival in excess of 8 months, and overall survival at 2 years. So this was really, for a group of patients that have very limited options, where standard of care offers a 1% response rate, we were thrilled with that result, and it did lead to an FDA approval. Now, one of the things that we were also fortunate to see was just an excellent tolerability, so there were very few adverse events that led to treatment discontinuation. And I'll point out that these types of adverse events that led to discontinuation include things like COVID-19 and unrelated to the actual drugs. And there were no deaths related to adverse events. And here's the toxicity profile for the tucatinib-trastuzumab combination. The most, that purple color is the grade 3, so those are the more severe adverse events. You can see most of the adverse events were grade 1 or 2 and fairly easy to manage with routine supportive care. And then the question is, are there certain groups of patients that benefit more or less from the tucatinib-trastuzumab combination? Our initial experience would suggest that age didn't have a big impact. ECOG performance status also didn't. Region of the world didn't. There may be a hint towards more activity for a left-sided compared to a right-sided primary, though we just didn't have enough numbers to really definitively conclude anything here. Now, I mentioned this question of blood NGS testing. When we took all the blood and tissue, looked at it centrally, and looked at how tucatinib and trastuzumab did using these central tests, IHC in fish, tissue NGS, and a blood NGS panel, and you can see that really these central tests show that IHC in fish really can discriminate a responder versus a non-responder. But if you look at blood NGS, progression-free survival is actually longer in a, quote, negative compared to a positive. Why would that be? Well, because if you notice, I called it ND, not negative. ND means not detected. That means that's a group of patients who are either true negative, meaning they wouldn't benefit, together with patients who are HER2 positive, but it wasn't seen in the blood, which is why I say go test tissue if your blood is showing it not detected. So not only do we have tucatinib trastuzumab, if you look in our guidelines, we have trastuzumab pertuzumab. There have been a significant number of international studies looking at that combination. That has a response rate closer to around 30%, PFS around 4 months. I think one of the limitations of this combination is just that it's, because it's two antibodies, it doesn't cross the blood-brain barrier, so the patients have a little bit greater risk of a central nervous system failure with that particular regimen in colon cancer, but otherwise it's a great regimen, very well tolerated by most patients. Now let's move to the other, what I consider major, significant, newly FDA-approved option for HER2-positive colon cancer, that's trastuzumab deruxtecan. We first learned about this from Destiny CRC01, which took patients who were HER2 positive, much like Mountaineer. They had progressed on at least 2 or more regimens. And all of these patients had HER2 positive disease by central testing using IHC in fish. And they looked at 3 cohorts, A, B, and C. B was an intermediate expressing, and C was a low expressing. Cohort A is where all the activity was seen. That's a classical HER2 positive patient, IHC 3+ or 2+ fish positive. Primary endpoint here was objective response rate, and the key thing is this was a very active drug in the initial experience, 45% response rate. The progression-free survival was around 5.5 months, and so, very impressive data. Now, one of the things you'll note here is if you look at those bars on the waterfall plot, you can see that almost all the responders had IHC 3+ disease. That's something you'll want to pay attention to, I guess, when you hear all of us talk about trastuzumab deruxtecan. Now, across the hallway, they're talking about managing HER2-positive breast cancer, and they're going to have a totally different story to tell. They're going to talk about these wonderful responses in these low and intermediate expressing HER2-positive patients. We don't have the same success story in colon cancer, at least. Only 1 response out of this first experience. And in fact, when you look at these progression-free survival and overall survival curves, you can see the progression-free survival in the low and intermediate expressing HER2 patients was about 2 months. All that benefit was over in the HER2 IHC 2+ fish positive or IHC 3+. So this is really a drug that appears to have a very select group of patients that it's very active in. Yelena already did a great job of explaining the safety considerations with this drug. It has the classic nausea, myelosuppression you'd expect, but one of the concerns with this study was there were 3 deaths out of 86 patients related to the ILD, pneumonitis signal, and one of the concerns was maybe they picked the wrong dose. It was this gastric dose at 6.4 mg per kg. Should they use the breast dose? So that's what inspired the Destiny CRC02 trial, which is shown here. This reported out last year. This randomized patients to the breast dose or the gastric dose, and with an idea to what's the most optimal dose for both safety and efficacy. And same population, colon cancer, central HER2 positive. And we see actually that there was a higher response rate, numerically higher at least, higher response rate in the lower breast dose compared to the higher gastric dose. Additionally, progression-free survival, pretty similar, maybe even a slight edge to the lower dose. And I would say that this basically drives home the point that the breast dose, 5.4 mg per kg, is the preferred dose for patients with HER2-positive colon cancer. And I'll also point out that we know a lot about this drug now in colon cancer. I really love this subgroup analysis because it tells you everything you need to know about this drug. IHC3+ really drives almost all the benefit here. You can see 47% response rate in IHC3+, but only 1 response out of 18 patients in a 2+ FISH-positive group of patients. RAS status appears not to matter as much. So RAS drives resistance to drugs like trastuzumab, pertuzumab, but not this drug. So you can give this to a patient who has both a RAS mutation and HER2-positive. And then it does not matter if this patient has progressed on prior anti-HER2 therapy. We still see significant benefit in those patients who've maybe progressed on a regimen like tucatinib, trastuzumab. You still see really closely similar response rates, whether they've progressed on prior HER2 or not. So let's finish up with this case. This is a 60-year-old woman, former smoker, no other underlying illnesses, and presented with weight loss and weakness and fatigue. PET scan showed a possible bone metastasis, but unfortunately she had both a sigmoid colon primary and liver metastases. They did the standard of care testing at the time, which was KRAS, NRAS, which was all wild type, and proficient mismatch repair. So for those of you who are watching and paying attention a little earlier, you'd say, oh, this sounds like a HER2-positive patient, right? It's got all those markers, left-sided, RAS wild type. And, well, we'll find out more. So this patient went on to get the standard of care at the time, which was FOLFOX and panitumumab, had a nice response initially, but around December 2019, we see, once again, that bright spot in the lumbar spine. And so this patient ended up getting resection after that nice response, and then recurred. So, you know, at recurrence, they ran NGS that included HER2, and the patient had HER2-positive disease. And so the patient ended up getting second-line FOLFIRI bevacizumab, but then went on a trial with dual HER2 blockade and had a partial response before eventually progressing. So I'd say for this patient with colorectal cancer, earlier HER2 testing would have been nice to know. We do associate this with resistance to anti-EGFR therapies. Might have even presented an opportunity to bring an anti-HER2 regimen earlier because the patient had to wait all the way through those lines of therapy to get access to an anti-HER2 therapy. And there are trials ongoing to bring these therapies up into first line. And so we see how those report out. Now, if you could answer this on your iPad, this question, I'll move on to the summary. And just some final thoughts. HER2 amplification or overexpression, I think, as you can see, is an actionable target for HER2-positive colon cancer. And I think the key thing is to test for it. And we know that there's a number of active therapies out there. We've got tucatinib trastuzumab, which is FDA approved. We've got trastuzumab deruxtecan, which is also FDA approved for IHC3+. And ultimately, we'll need to decide as clinicians how we sequence these therapies, and that will all depend on kind of clinical and genomic characteristics, efficacy, and tolerability profiles. So with that, Dr. Pant.
Oh. Well, thank you so much, Yelena and John. It's obviously, I've got big shoes to fill coming up after them, so I wish I was the first speaker, but I was the last when I was looking at the slides. But I'll try to keep you guys engaged. I know this is a long first day at ASCO, so we'll try to kind of, talk about this cancer, which people really did not talk about a decade back. And now we've got newer targets, newer sequencing. So it's really where we are seeing all this progress really fast. And I want to say one thing, we talked about, the advocacy group. So one thing which has really made a difference in this disease is the advocacy in this, and specifically the Cholangiocarcinoma Foundation, Stacy, Melinda, and Juan, who are right there. I've gone to a lot of meetings, and I had the distinct pleasure to go to the Cholangiocarcinoma Foundation meeting. It's in beautiful Utah, so beautiful place. Great city, but the most important thing is it's a very unique meeting. It's got physicians, clinicians, researchers, basic science researchers, and then it has patients. So I was presenting a poster this year at the Cholangiocarcinoma Foundation meeting, and so many patients came up to me and talked to me. They were like, hey, I have this mutation. And I was presenting on HER2, and they said, oh, I have the HER2 amplification. And it's a really unique meeting where you really get to talk to the patients, personally interact with them, and that makes it really special. So if anybody, I think that's a meeting that everyone should definitely, whether they're, interested in cholangiocarcinoma or not, should definitely try and attend to really look at that ecosystem which has been built in the cholangiocarcinoma community, which I think has led to a lot of these advances. So thank you for that, Stacy. Now I'll talk a little bit about, biliary tract cancers. So it's important to remember cholangiocarcinoma is not one cancer, it's different cancers. So broadly, you have extrahepatic cholangiocarcinoma outside the liver, intrahepatic cholangiocarcinoma in the liver, and gallbladder cancer. And all these are very different because they are in different parts of the world. The prevalence matters of these diseases, and specifically, they're very different genomically. So really, when you're testing them, it's very important to know, even though it's a rare disease, right? You still have to parse through the different types of cholangiocarcinomas. They're all not the same. That's important. So what are the current shortcomings in the management of advanced biliary tract cancers? So one of the things, common theme is if you do not test, you will not find. So we really need to do next-gen sequencing on all our patients with biliary tract cancers because they have targetable alterations. Whether they can go on an approved drugs, which they are for different mutations and fusions like FGFR and IDH1, or if they can go on clinical trial and now for HER2, or they can go on clinical trials for different, for different mutations and alterations. So it's very important, but if you do not test, you will not find. So that's the most important thing, that you test these patients. What are the other issues? So this was an assessment, which was a survey sent out to providers about cholangiocarcinoma. So as you can see, 85% of patients are initiated on gemcitabine-based chemotherapy. Most commonly now is gemcitabine, cisplatin, durvalumab. Pembrolizumab also has been added to the armamentarium. But after the first line, this fall-off is really acute. So only 46% of patients initiated second-line therapy, predominantly with 5-FU-based chemotherapies. Few patients, very few, 17%, moved to the third line of treatment. And we really need to move this curve with more patients being treated in second line and third line, specifically if they're molecularly targeted. And why did I say this? These are different cancers. Overall, this is a busy slide, but overall, you have to remember intrahepatic cholangiocarcinoma are your FGFR2 fusions and your IDH1 mutations. That's where you can find these. Extrahepatic cholangiocarcinoma, you have HER2 and you have KRAS. And gallbladder cancer, interestingly, if you really look at the HER2, because that's the focus of the talk, intrahepatic about 5%, and these are kind of general numbers. Extrahepatic 10 to 15%, and gallbladder can be 25 to 30%. So it can be that high in gallbladder cancers. So really testing does matter in all these patients. This is chemotherapy, so second-line chemotherapy through the ABC06 trial. As you can see, the median progression-free survival was about 4 months, and the overall response rate for these patients in this second-line unselected group who got FOLFOX was 5%. And that's important to remember because we are going to talk about the response rates to HER2-directed therapy in the coming slides. So 5%. And about prognosis, and John talked about this before. In colorectal, we're not sure. In biliary tract cancers, we do have hints. Again, not big data sets, but we do have hints that it's probably a little bit more aggressive tumor, maybe similar to breast. That's what I think about it, because when you looked at these curves, which was the breast cancer HER2 positivity way back in the 90s, you could see that the patients who were HER2 positive disease, prognostically, they did not do as well. And if you can see, this is from gallbladder cancers, patients with resected gallbladder cancer. And as you can see, the ones who are HER2 positive, they did fairly poorly, small numbers. The other important thing is, Remember, I told you 30% of patients with gallbladder cancer, up to 30% can have HER2 positivity. And gallbladder cancer is a different disease in a way than intrahepatic cholangiocarcinoma. Intrahepatic cholangiocarcinoma, it's maybe slower progressing. Gallbladder is a little bit more rapidly progressing. Patients present with pain. They present with kind of these symptoms that we see. And it's a little bit more aggressive than your other tumors. And maybe HER2 is driving some of that percentage. So one of the first trials, and a lot of the data in HER2 biliary tract cancers is part of basket trials. Again, rare cancer, rarer mutation. So most of the data that I'm going to present are parts of basket trials. This was one of the first ones, the My Pathway, which was trastuzumab with pertuzumab. And in this, there were 39 patients who were eligible for efficacy analysis, and the overall response rate in these patients previously treated was 29%, so about close to 30%. Again, we talked about PDX. This was the HERB trial. Again, this was run in Japan. In this, again, there were about 30 patients, which were patients with HER2 positive disease. The response rate in these patients was about 36.4%. So kind of in that ballpark for different, for different agents. So pertuzumab, trastuzumab, and then this is the ADC. And this is the trial that recently was presented in ASCO last year, and this gained approval recently for these patients. So this was the Destiny Pan-Tumor 02, which had broad activity for HER2, different malignancies, including HER2 positive BTC. And as John pointed out, that majority of the responses out here were HER2 3+. So again, the 3+, very different than 2+. We'd see these high responses in 3+. So if you look at this biliary tract cancer, they were, overall 41 patients, 22% response rate. Almost all the response in these patients was driven by HER2 IHC 3+. As you can see, a good response, 56.3%, all driven by IHC 3+ patients. And there was a tumor-agnostic approval for TDX. Again, the updated NCCN guidelines for second or subsequent therapy for HER2-positive tumors. Again, patients who are 3+. And also, this has gained accelerated FDA approval for all adult patients with unresectable or metastatic HER2-positive 3+ disease solid tumors who have received prior systemic therapy and have no satisfactory alternative treatment options. And ASCO 2024, this data is going to be further presented tomorrow, June 1st at 1:30 p.m., abstract 4090. So you can look at some of the more data with this disease and with this drug in this disease. And I want to point out something which Yelena said before is about this, and John, about the side effects. Again, you do have these side effects with this drug. For what I see is it's not a benign drug, so patients do get fatigue, they do get anorexia, they do get nausea. So you really have to, and interstitial lung diseases, again, is what people talk about, but really when the patients, majority of them, they do get these side effects, and you really have to, really follow aggressive supportive care to get them through this therapy. That's very, very important. Those are the main ones that we see. Coming to tucatinib and trastuzumab, we talked about it a little bit in the colorectal space. Again, this was presented last year again. This was, again, part of a basket trial. As you can see, there were 29 patients. The overall response rate was 46.7%, and the median duration response was 6 months. So, and the main side effect, as John said before, is diarrhea. That's, you really have to manage the diarrhea. The majority is grade 1 and 2. Coming to zanidatamab, kind of one of the newer kids in the block, and this was, again, a basket trial of 22 patients. So just like John said, I have a story about this. I won't take much of your time, but this is pre-pandemic. I was in the phase 1 department in MD Anderson. I had a patient with gallbladder cancer, presented with pain. She was on gemcitabine-based therapy. We tested her. She was HER2 positive. We had this trial open. We gave zanidatamab to that patient. And it was very interesting. The first day when I started the drug, she was doubled over in pain. She said, pain had risen, as we see in gallbladder cancer patients. And then a month later, when was the first follow-up on this trial, the patient just walked into my clinic. And I was like, man, that was so different. Like, she was really doubled up in pain. She was smiling. She was talking about her gardening, about bothering her daughters. Her daughters used to come with her. I said, this is something different. I really haven't seen this in BTC cancer patients with this. So 2019 ASCO, we went to the company, and they said, I don't know, this looks okay, but small patient population, let's see, we're a small company. So every time we had a response at MD Anderson, we would go to the company. We would be like, hey, look, there's another response. And finally, in ASCO 2019, pre-pandemic, they finally decided to do the global phase 2b trial that I'll show you guys a little bit of data. So really, you can learn a lot from the patients. So this was another cool kind of example of really, companies and us working together. But this was the phase 1 trial in which we saw an overall response rate of 38% in the end. And this led to this trial, the Horizon BTC-01, presented in ASCO last year. It's a global phase 2b trial of zanidatamab monotherapy in HER2-amplified BTC. This drug, after frontline, whatever they could have received chemotherapy, HER2, they had to be 2+ or 3+-ish positive, inside hybridization positive. Patient got zani day 1 and day 15. Primary endpoint was overall response rate. We enrolled 80 patients which were BTC 2+ and 3+, globally, and we had a response rate of 41.3%. And it was not just the response rate. The important thing was the duration of response on this trial was 12.9 months. So when you look at it clinically, patients can get a big response, and if they progress with their next scan, I don't think it helps them a lot. You want that duration response, you want that durability of response to really look at if the drug is really having an impact on the patient. So really, and like I said, the median time to first response, just like my patient, was about 1.8 months, so relatively short. This drug is working in the patients. Literally, even you can see them in 2 weeks, and if their symptomatology has gone down, it's working. And this was adverse events of special interest, diarrhea, and cardiac events. So diarrhea, again, mostly grade 1, grade 2, very tolerable, with antidiarrheal medications. This was presented last year as a follow-up, quality of life outcomes. If you got a response, you had a better quality of life outcome. That's the bottom line for this abstract. But the updated results of the overall, we are reporting the overall survival analysis. Now we have a median follow-up of 22 months, presenting it tomorrow, 1:30, again in the poster session. So it'll we'll show you about our duration response on this drug and updated overall survival, which we did not have last year. This is another interesting, now moving on to chemo combinations. This was a study which was actually done in a single center in India called Tata's. Amazing center. If you ever get a chance to go to Mumbai in India, they're doing just fantastic work. Gallbladder cancer, interestingly, if folks want to know, big prevalence in India, northern part of India, in what is called the Gangetic Plain. It's also in in Asia, and also in the mountainous regions of Chile. So different, interesting kind of prevalence in different parts of the world. So they enrolled this in the single center, or just in the Tatas, they enrolled these patients, 90 patients with HER2 positive, which would take me about 5 years to accrue one more in the US, but 90 patients, and they accrued this with trastuzumab, with gemcitabine and cisplatin, and this was the result. The 12-month overall survival, the median overall survival was 9.96 months. Doesn't sound like a lot, but gallbladder cancer has worse prognosis than other biliary tract cancer. So that's very important to remember about this trial, about the nuance of this trial. This was another smaller trial of 34 patients, FOLFOX with trastuzumab, again, overall response rate of about 30%, with a median progression-free survival of 5.1 months, so it can be combined. And there's a lot happening in this field, so stay tuned for the next 3 to 6 months. There's just a lot happening in this field, so I'm really happy that that biliary tract cancer, is finally having its moment in the sun. So that's really amazing to see in this disease. And I'll end with a case. So patient, 60-year-old man, initiated on gemcitabine, cisplatin, durvalumab. Subsequent NGS testing revealed HER2 amplification. And how do you approach targeted selection for this patient? And how do you counsel on the potential AEs? Thank you so much. I appreciate it.
This was an excellent overview. So maybe, because you brought up your experience ex-US, there's a lot of good questions, and thank you for your participation. What do you think, and Subham, maybe I'll start with you. What do you do with the economic and geographical barriers to receive HER2-targeted agents? I mean, certainly, even in Eastern Europe and places like this, I hear that people don't test for HER2 because they don't have access in gastric to trastuzumab. What's your experience? I mean, you mentioned this.
Yeah. Thanks for the easy question,
Yeah.
first one, Yelena. But yeah, so I think that's a real, the thing is we can approve all the drugs we want, but if there's no access to it in majority of the world, then we are leaving a large chunk of the world out. I think, at least my experience, when I went to India last year, we went part of this with cholangiocarcinoma foundation, is that the HER2 testing is, at least where the prevalence is high, because they know to test for gallbladder cancer because it's so high, they were testing with almost all their patients for HER2 IHC, which is a cheap test, can be done. But it was being done, and they had standardized it.
The setting of a trial.
And yeah, but they're now doing it for essentially majority of patients because it doesn't cost them a lot to do that HER2 testing, at least in India. So that's the only that's the only outlook I have for any country other than the US. So I don't know if you have anything else.
Yeah, I mean, so the good news is it will not break the bank to do immunohistochemistry. That is one of the more accessible tests. So if you're in a resource-limited environment, immunohistochemistry is pretty easy and low cost. And look, there's data. If you look at colon, the very first therapy that showed benefit was lapatinib with trastuzumab, and landed in our guidelines. Those are two off-patent options, right? If you're in a resource-limited environment. So fortunately, we're in the United States where we get a lot of assistance from our biopharma partners to support patients with copays and other burdens. So I think that while I do treat a broad range of patients from all different types of economic backgrounds, I have not had a hard time with accessing even some of our newest FDA-approved agents, drugs like trastuzumab deruxtecan. It has not been a problem, knock on wood.
In the United States.
In my clinic, yeah, it's not been a problem.
Great.
And so I'm thankful for the companies that are willing to support our patients who struggle.
So maybe you can take the next set of tucatinib questions. Why, in combination therapies type of thing, why would capecitabine be so important in breast in combination with tucatinib but not colon? And also, would you ever treat a RAS mutant case with tucatinib, or would you only give TDXT in that case?
Yes, those are really good questions. So, when I initially wrote that Mountaineer trial, I was looking at patients who were heavily refractory to chemotherapy, 5FU, Xeloda. Patients had already progressed on probably multiple lines of 5FU or Xeloda-based treatment. So, I'm looking at a group of patients that is heavily pretreated, not a lot of reserve to handle the side effects of cytotoxic chemotherapy, and I really wanted it to be exceptionally well tolerated. And I felt like the preclinical data supported a chemotherapy-free regimen for patients, tucatinib, trastuzumab. And fortunately, the data bore out. And I think it was always meant to be a quality of life regimen. The question is, how much are we losing by dropping the capecitabine? And in truth, there might be small incremental benefit, but I don't think that the toxicity of adding capecitabine to that group of patients is worth it. Now, that's my own personal view, but it's never been looked at. Now, the other question is what to do with a RAS mutated HER2 positive patient. How should we manage that? I very deliberately excluded patients with RAS mutations from Mountaineer because RAS mutations are really designed to rescue tumor cells from selective inhibition, whether it be EGFR for a RAS wild type. And the data from my pathway suggested RAS mutations really did drive resistance in a very potent way, and I had no reason to think that that would be any different with tucatinib trastuzumab. Now, on the other hand, the antibody-drug conjugate trastuzumab deruxtecan, because it has a cytotoxic chemotherapy component to it, it just needs to get in the cell. It doesn't have to shut down a pathway, so that RAS mutation is less relevant to that drug. So I would favor trastuzumab deruxtecan for that type of patient with a RAS mutation or HER2 positive.
Great. Agreed. Shubham, what about for biliary tract cancer? Do you test patients in first line? Do you use IHC and GS? Do you want to use anti-HER2 in first line or second line? And maybe I'll throw in a sprinkle of RAS also on that. Yeah. Tell us how would you do biomarker testing in biliary tract cancer?
Yeah, so I think everybody should get, at least in the U.S., NGS testing for biliary tract cancers. I think it's important, just not HER2, which is new, but FGFR fusions. We have 2 approved drugs for that. IDH1 mutation, and then now HER2, and then obviously looking for clinical trials for patients. It's important. Now we have a whole slew of RAS inhibitors. So last year we had reported out on a Crystal-1, which was a data set for a KRAS G12C inhibitor, adagrasib, and it's again not very common, but common in colorectal, lung, not very common in pancreatic and biliary tract cancers. But we looked at our data set, which was 60 patients with panc, biliary, and other cancers in this basket, and we saw pretty decent response to the G12C inhibitors, adagrasib. It's now on the NCCN guidelines, updated NCCN guidelines. So there's also RET, there's NTRK. So BTC has the
HER2 BTC, does it also have KRAS amplifications or mutations?
Yeah, that's a good question. So the HER2, they can have, but it's a smaller percentage.
And would you not target HER2 if you saw KRAS? What would you go after?
So when we did we're going to look at the biomarker for the Zani data
so we
that we did.
Yet.
Yeah,
okay.
So the others have been like smaller baskets, so not a lot of information out there.
Yeah.
But I think it's going to follow with the trastuzumab, pertuzumab. If you give it, I think it's going to kind of follow the same path of if you have a RAS mutation, that's the driver. I think in the ADC, it might not matter, like we talked about trastuzumab deruxtecan.
Right.
There, the RAS may not matter. It may be similar in biliary as it's in colorectal on that the trastuzumab, pertuzumab may matter, the RAS status may matter, but this may not. We'll look at the Zani data. We're evaluating the biomarkers right now.
Yeah, and so there's a lot of questions about what's the appropriate test, and I think in first-line setting, the quickest test is the most appropriate, IHC. We don't wait for NGS typically, but certainly for colon cases, we need to know the RAS status, so a limited RAS panel, or even just getting ctDNA test also may be important, especially as you're bringing these agents to first line. I think questions came up about HER2 mutations as opposed to amplification. There's certainly pan-tumor approval, and HER2 long mutations do occur, and T-DXd has great data there. In gastric in particular, these mutations are usually on the internal domain and typically have not been very successful in being targeted. We don't have data specifically for T-DXd in those patients, but typically if they're not amplified, then the target is not as effective for HER2.
Same thing,
yeah. So, and then in terms of copy number alterations and what is IHC3 plus is equivalent to, that's a great question. And I think not every IHC3 plus is created the same. It's in terms of tumor content and the quality, and also the how if it's very focal positivity in 5% of cells, you won't even see copy number alterations. You will see basically very flat, sub-clonal amplifications. And if you do have high level of amplification, usually it's very obvious. As I said, IHC3 plus patients are usually only if they have really homogeneous expression.
When we looked at the Zani data, about 52% HER2 3 plus was 52% response rate, the higher than 41. 2 plus, again, we had it was a 5% response rate, but we did have stable diseases with the didn't reach a 30%, but they got to a 10%, 20%. So we did see some of that also. So I think even in the 2 plus, HER2 targeting, might work in some. Again, it could be maybe, how they've stained it, right? It could be a 3 plus, but you're looking at a 2 plus.
So in the last few minutes, maybe we can talk a little bit about the right and left side colon, and also, prioritizing BRAF versus HER2 as targets, and how do you think about it?
Yeah, I thought that was some of these questions are really,
really
putting us
very
in a
good.
bind.
Yeah.
Right. So I will tell you, I've see a lot of molecular profiles. I also run our molecular tumor board. I'm trying to think. I'm aware of a couple of cases over the last 5 to 10 years that are both BRAF V600E mutant and HER2 positive. They tend to be right-sided. The question is, what would you target first if you had both a BRAF and a HER2? I would actually use the immunohistochemistry in that setting. So I think we can rule out using drugs like tucatinib, trastuzumab, and more of the selective targeted therapies there. I think we would not expect much benefit. If the immunohistochemistry stain is 3+, I really like trastuzumab deruxtecan for an IHC 3+ HER2 positive cancer, regardless of whether there's a BRAF mutation. If it's IHC 2+, I would go with the anti-BRAF strategy. That would be how I kind of parse that, but I don't get tested on that one very often.
Well, you passed the test.
Yeah.
Great discussion. Thank you so much for all your insights. We learned a lot, and thank you for your participation. And have a great rest of your ASCO, and hope to see you at the next event. Thank you.